Good morning, students. So, our next topic discussion: research areas of biotechnology. In last class, we have discussed about biotechnological applications. Applications of biotechnology. So, applications of biotechnology in that how biotechnology are useful in various fields of human mankind. So, after that, research areas of biotechnology. So there are three critical research areas of biotechnology. Three critical research areas. Critical areas of biotechnology. Three critical research areas of biotechnology. Those are, first one is providing best catalyst. Providing best catalyst. Providing best catalyst. Best catalyst in the form of improved organisms or genetically modified organisms, which may be microbes, or enzymes, There are three critical areas of biotechnology. Biotechnology mainly research process carried in three areas. In those three areas, biotechnological applications and or researches or experiments are widely our scientists are doing. Among those, first one is providing best catalyst. Providing best catalyst. Catalyst means to speed up the work. See, last class we have discussed cloning of desired gene through bacteria. It's a time-taking process. Nearly 1 month time required. But PCR, within few hours, billions of copies produced. PCR is a biotechnological application. Yesterday we discussed that diagnosis, PCR and ELISA tests are used to detect or diagnose certain disorders. So biotechnology means speed up the biological experiments, not only providing best catalyst. That catalyst may be in the form of microorganisms or improved organisms or genetically modified organisms. That's maybe a microbes. Or enzymes. So enzymes are used to speed up the reactions. Okay, so PCR technique we have used Taq polymerase enzyme, fast of the replication process and produce quickly millions of copies. Okay, so that is the first research area of biotechnology, providing the best catalyst in the form of genetically modified organisms. That's maybe microbes or enzymes. Second research area, creating optimal conditions. Creating optimal conditions. Creating optimal conditions through genetic engineering to act as effectively. Creating optimal conditions through genetic engineering to the catalyst to perform their work effectively to the catalyst. Creating optimal conditions. To genetic engineering to the catalyst to become active. Okay, so to the catalyst, optimal conditions are created. The optimal conditions, catalyst work effectively to make their work effective. Optimal conditions should not exceed more temperature, should not decrease temperature. Oxygen and carbon dioxide content, nutrient content, pH content, vitamins content, all whatever may be the conditions are required to the microbes or enzymes, whatever it may be. So optimal should not exceed more, should not get it. Optimal conditions are required. Optimal conditions, creating optimal conditions through genetic engineering to the catalyst. Okay. Third one is downstream processing. Downstream processing technologies. Already we have discussed that. Downstream processing technology refers to the separation and purification of desirable products and organic compounds. Not only protein products, some of the organic compounds, some of the enzymes, medicines, etc. are prepared. So whatever may be the products are biologically synthesized, prepared. The main step is the downstream processing technologies. That means separation and purification of food, proteins and other organic compounds. Another organic compounds. So these three are critical research areas of biotechnology. You may be get out, sir. In last class we have discussed about recombinant DNA, where it was, it is a major for biotechnology now. Yes, it was a major construction of recombinant DNA technology is a major process in biotechnology. That is what in first step only is there. Okay, providing best catalyst in the form of genetically modified organism. Genetically modified organism means what? Foreign gene. Foreign gene means transgene, desired gene. When introduced into the normal organism, then it becomes genetically modified organism. Okay, these three critical research areas of biotechnology are very important. Okay, that's yes. Okay, next topic. In this chapter, we will discuss about biotechnology, how this is helpful to mankind in production of agricultural products, foods, medicines, and other enzymes. Among those, first of all, how biotechnology is useful in agriculture. Okay, so biotechnological applications. Biotechnological applications in biotechnological applications in agriculture. Biotechnological applications in agriculture. Okay, biotechnological applications in agriculture. First of all, there are three techniques helped to increase the food products. Increase the food products carried out by three important steps. Through three important steps. Those three important steps are three important techniques. Increase the food production can be brought by three important means. Three important means techniques. Those are, first one is agrochemical based agriculture. Agrochemical based agriculture. Second one is agriculture. And third one is genetically modified crops. Okay, agrochemical based agriculture. Agrochemical based agriculture made up of plant breeding techniques. Agrochemical based agriculture made up of plant breeding techniques. Plant breeding techniques. Plant breeding techniques like hybridization. Okay, agrochemical based agriculture mainly made up of plant breeding techniques like hybridization. What is meaning of hybridization? So, cross breeding in between superior plants. Okay, so agrochemical based agriculture is made up of plant breeding techniques like hybridization. Hybridization, first one is along with the use of agrochemicals. Use of agrochemicals. What is agrochemicals? Pesticides, fertilizers only called agrochemicals. Plant breeding techniques like hybridization along with the use of agrochemicals. Better management practices, and improved crop varieties. These are all the techniques. Agrochemical based agriculture is made up of plant breeding techniques like hybridization, agrochemical based along with the use of agrochemicals, better management practices, and improved crop varieties, which brought revolution, green revolution and increased food supplies enormously for ever growing population. There needs to be produced crop production for ever growing population without getting scarcity, without getting deficiency. Okay, so that is called agrochemical based agriculture. So agrochemical based agriculture means what? First one is hybridization techniques. Next one is use of agrochemicals. Use of agrochemicals means what? Fertilizers, pesticides, insecticides, weedicides. And better management practices. Better management practices. Management practices means proper irrigation, proper ventilation, proper weedicides removal, and proper time everything supply is called better management practice. And at the same time, we have to use improved crop varieties. So best plant varieties we have to use. So these are all the techniques brought green revolution. Increase the food supply in vigorously, enormously. It would sufficient for our growing population. There is a need to supply excess amount of food yield for ever growing population. Okay, that is called agrochemical based agriculture. Next, organic agriculture. The main aim of organic agriculture: reduce the need for fertilizers and pesticides. Pesticides. Okay, so the main aim of organic agriculture is reduce the need for fertilizers and pesticides. Okay, so only they will use biomanure. With the help of biomanure and disease-resistant varieties, they have to improve the food supply. Okay, the main aim of reduce the chemical fertilizers and pesticides, which are not only harmful to the mankind, but also pollute the environment. pollute, pollute the environment, pollute the environment. Not only harmful to the human mankind, also it pollute to environment. So, but here organic based agriculture is highly expensive. Though it is very expensive, it also requires a very good knowledge of pest, pest or pathogen management. Pathogen management. Agrochemical based agriculture is highly expensive. Though it is expensive, it requires very good knowledge of best, very good knowledge of pest and pathogen management. Okay, so frequently, what kind of at the initial stage, what kind of pathogens we adopt to remove them? What precautions we have to take? At the time of crop, at the time of fruit giving time, crop giving time, after crop harvesting, what kind of pathogens commonly adopt those crops? So that kind of knowledge we will have them. So that is called organic agriculture. Organic based agriculture means what? Without using fertilizers and pesticides, only biomanure they will use and produce crop and get excess amount of yield. Okay, so the main aim of organic based agriculture is reduce the need for fertilizers and pesticides, which are not only harmful to the mankind but also pollute a valuable environment. Okay, so it is a highly expensive and required a very good knowledge of pest or pathogen management. Genetically modified crops have made a way. Genetically modified crops have made as an alternative pathway for the production of yield or crop to genetically modified plants. Through genetically modified plants. So the genetically modified plants reduce the dependence on fertilizers and pesticides. Reduce the dependence on fertilizers and pesticides. That is genetically modified crops. The main aim of genetically modified crops, production of food, production of food materials, production of food materials through genetically modified crops, genetically modified plants, which reduce the dependence on fertilizers and pesticides, which means genetically modified crops will withstand whatever may be the natural calamities occurs. If any bacteria, virus, pathogens attack, nothing will happen to the genetically modified crops. You may be get out. Why, sir? Why nothing happens to the genetically modified plants? For that question answer, we will get to the next topic. Okay, so genetically modified crops, they can produce huge quantity of food materials. At the same time, they reduce the dependence on fertilizers and pesticides. Fertilizers and pesticides. That is called important plant breeding. So these three steps, three techniques, that is agrochemical based agriculture, organic agriculture, genetically modified crops. So these three modern techniques have two unique advantages over traditional plant breeding techniques. These modern techniques have two unique advantages. Two unique advantages over traditional plant breeding techniques. Those advantages now is that advantages of modern techniques. Unique advantages of modern. Okay, see. First, first technique. See, in modern techniques, any gene can be synthesized artificially. In modern techniques, any gene can be synthesized in laboratory or from the plant can be collected and inserted into another plant. That is called first advantage. Okay, so in modern techniques, any gene, the desired gene can be synthesized or collected from any another unrelated plant or animal or any other organisms in the laboratory and inserted into the plant. Okay, so first unique advantage. In modern techniques, any gene can be synthesized in laboratory and inserted into the plants. Not only recombinant DNA cloning of factor, the plant which is growing, in one particular place, the plant contains superior characters, giving more crop, disease resistant variety. Any climatic conditions it is proliferating. In any climatic conditions it is surviving. From that kind of plant, they collected a gene. This is called transgene, foreign gene, or alien piece of DNA or gene of our interest. That gene is collected. The same gene may be synthesized in the laboratory or through cloning. The same gene is multiplied and inserted into the. That is the modern technique. In modern technique, any gene can be synthesized in the laboratory or collected or multiplied through cloning and inserted into the plants can be created desired traits. Plants can be created desired plants since introduction of transgene. See here. So through cloning we have multiplied gene, or in the laboratory we have multiplied gene. The gene is inserted into the plant. The plants can be created desirable traits. What are the traits we want? Those traits we can be created. Since only introduction of transgene, nothing but foreign gene. Transgene, nothing but foreign gene. Here, definition of transgene. Definition of transgene. Definition of transgene. Definition of transgene. Transgene, it is a foreign gene. It is a foreign gene which can be collected from unrelated plant, animal, or any organism and introduced into lesser superior plant. Make them to superior. That is called transgene. Okay, so the gene which is inserted into the plants. In those plants, we can bring desired traits. So this gene is collected from the superior plant. In the superior plant, what the characters are there, those characters we can bring in another plant. Plants with desirable traits we can be created. Since only transgene when we are inserted into the plant. These two are unique advantages. Here you never get down to transgene. Transgene means foreign gene. Foreign gene or alien piece of DNA or gene of our interest. Okay, foreign gene, the plant or animal which is growing superiorly, which have superior characters from those plants or animals will collect the DNA. The DNA is multiplied and inserted into another plants. Such type of gene is called transgene. Simply within one word, it is a foreign gene is called transgene, which is collected from unrelated organisms. Unrelated organism or plant or trans. Genetically modified crops. So genetically modified crops are genetically modified organisms. Genetically modified organisms. Genetically modified organisms which contains a foreign gene, which contains foreign gene in their body, in their plant body. Such organisms are called genetically modified organisms are transgenic, transgenic organisms. Transgenic organisms. Genetically modified organisms which contains a foreign gene. Such type of organisms are called genetically modified organisms are transgenic organisms. Transgenic organisms could be bacteria, fungi, plants, or animals. Okay, that's so genetically modified organisms or transgenic organisms could be bacteria. Bacteria is available in the form of fungi, available in the form of plants or animals. Next here, genetically modified organisms are useful in many ways. So what are those ways? Already we have discussed yesterday only in medicine, bioremediation, in pharmaceutical industry, waste treatment, energy production, in different streams. Genetically modified organisms are useful in many ways to the human mankind. Already being discussed earlier. Okay. So this is the information about biotechnological applications in agriculture. Next, advantages of transgenic crops or transgenic organisms. Transgenic plants, all are same. Transgenic crops or transgenic animals or transgenic plants, all are genetically modified organisms. Among those, first of all, we will see advantages of transgenic plants or genetically modified plants. So those genetically modified plants topic, next class we will see. Next class we will see. Today meeting session is finished. Get ready for next class. I am leaving the meeting.